Hello all, I am Pravina from first year MSc Botany. I am here to discuss with the topic structure, reproduction, physiological characteristics and uses of lichen thallus. Lichens are the plants with composite nature consisting of two dissimilar organisms, an algae and a fungus living in a symbiotic association. The algal partner is called phycobion and the fungal partner is called mycobion. These are the two words from the Greek and meaning phyco, algae, bio means life, myx means fungus, bio means life. The symbiotic association of algae and fungus in a lichen is as follows. The fungal partner occupies a major portion of the thallus and produces its own reproductive structures. The algal partner manufactures the carbohydrates through photosynthesis, which diffuses and is absorbed by the fungal partner. So, based on the morphological structure of the thalli, there are three types of lichen: crustose, foliose and fruticose. In this picture, we can see the crustose lichen, foliose lichen and fruticose. So, crustose lichen. The thallus is inconspicuous, flat and appears as a thin layer or crust on substratum like barks, stones, rocks, etc. Example, rhizocarpon, like that. Foliose. Foliose lichens, these are the leaf-like lichens where thallus is flat, horizontally spreading with lobes. Some parts of the thallus are attached with the substratum by means of hyphal outgrowth. The rhizines develop from the lower surface. Example, Parmelia. Fruticose. These are shrubby lichens where thalli are well developed, cylindrical, branched. Example, Usnea. Looking into the internal structure of thallus, based on the distribution of algal member inside the thallus, the lichens are divided into two types: homomerous and heteromerous. Homomerous, the fungal hyphae and the algal cells are more or less uniformly distributed throughout the thallus. The algal members belong to the cyanophyta. This type of orientation is found in the crustose lichen. Both the partners intermingle and form the thin outer protective layer. Example in Leptogium and Colema, etc. Heteromerous lichen. The thallus is differentiated into four distinct layers: the upper cortex, algal zone, medulla, and the lower cortex. Upper cortex is thick outermost protective covering made up of compactly arranged interwoven fungal hyphae, and the algal zone occurs just below the upper cortex. The algal cells are tangled by the loosely interwoven fungal hyphae. Medulla. The medulla is situated just below the algal zone, comprised of loosely interwoven thick-walled fungal hyphae with large space between them. The lower cortex, the lowermost layer of the thallus. This layer is composed of compactly arranged hyphae which may be arranged perpendicular to or parallel to the surface of the thallus. Some of the hyphae in the lower surface may extend downwards and penetrate the substratum, which help in anchorage, known as the rhizines. In addition to the fungal and the algal cells in the lichen thallus, we can see some other special vegetative structures. They are the breathing pores, Syphilia, Cephaloidea, Isidia and Soredia. So, breathing pores seen in foliose lichen, it is cone-shaped depressions for gas exchange. Syphilia. Aerating organs appears as a cup-shaped white spots on the thallus. Syphilia opens to medulla to enable gas exchange. Cephaloidea. Formed of algal cells enclosed by fungal hyphae of hard and dark gourd-like or wart-like internal or external swellings. Isidia. Papillated outgrowths formed on upper surface of the thallus function to increase the photosynthetic efficiency of lichen by increasing the surface area. Soredia. The round bud-like outgrowth on thallus or in pustule-like areas called soralia appears as greyish powder on surface. Reproduction. The lichens reproduce vegetative, asexual as well as sexual methods. Vegetative reproduction by fragmentation, isidia and soridia. Asexual reproduction by the formation of oidia, conidia and pycnospores. Sexual reproduction by ascospores and basilospores. Vegetative reproduction. The decaying of older parts and break into smaller parts, with the smaller parts later develop into the new lichen thallus. Soridia. Powdery propagules composed of fungal hyphae wrapped around cyanobacteria or green algae. Isidia. Outgrowths of the thallus surface consisting of both mycobiome and phycobiome. Sexual reproduction by oidia formation. Hyphae breaks up into the component cells or small pieces and develop into the spores. Conidia. Fungal components forms a conidia on conidiophores and it gets released from the lichen and forms a new mycelia. Pycnospores. Conidia formed in flask-shaped structure called pycnidia that are embedded in thallus. It is liberated and undergoes germination to form the fungal mycelium. Sexual reproduction by ascospores or basilospores. Ascospores are produced in the ascolichen as well as the basilospores are produced from the basilolichen. The male sex organ is called flask-shaped spermagonium, produces unicellular spermatia, where the female sex organ is carpogonium or ascogonium, differentiated into basal, oogonium and elongated trichogyne. The spermatia behave as male gametes. The spermatium, after liberating from the spermagonium, get attached with the trichogyne at the sticky projected part. On dissolution of the common wall, the nucleus of spermatium migrates into the carpogonium and fuses with the egg. Many ascogenous hyphae develop from the basal region of fertilized ascogonium. The binucleate penultimate cell of the ascogenous hyphae develop into the ascus. Both the nuclei of penultimate cell fuse and form diploid nucleus, which undergo first meiotic and then mitotic division, result in 8 haploid daughter nuclei, each haploid nucleus with some cytoplasm metamorphoses into ascospore. So this is a picture of the carpogonium and spermagonium seen here. The fruiting body called apothecary. The fruiting body in lichen is called apothecia or perithecia. Apothecia is a disc-shaped in asnia and perithecia is a flask-shaped fruiting bodies. It consists of asci, which in turn produce the ascospores. Structure of apothecia. The hymenium, the tissue containing the asci, is seated at the base of the peritheal cavity. Asci remain intermingled with some sterile hyphae, the paraphyses. Hypothecium is a tissue below hymenium and from which the hymenial elements develops. Acciput is the rim of apothecium. Epithecium is the layer above the asci consisting of paraphyses embedded in the gelatinous material. The apothecium consists of epithecium, paraphyses, hymenium, acciput and hypothecium, and the asci are produced in the epithecium. Economic importance of lichen. So, medicinal uses. Asnia, in the treatment of urinary complaints, asnic acid obtained from asnia is a broad spectrum of antibiotic. It is used in the preparation of wound healing ointments, and peltigera in the treatment of rabies. Protolycosteric acid obtained from lichens have anti-carcinogenic properties. Asnia evernia used as anti-strings in the hemorrhages. Cladonia pyxidata effective in the whooping cough. As food, species of Parmelia known as the rock flower used as a curry powder in India, and France lichens are used in coin factory for making chocolates and pastries. Cladonia rangifernia as a main food for reindeers, and Cetraria islandica are used as a powder made into cakes. Species of Evernia parmelia are edible. Industrial uses used in the brewing industry and distillation industries for the production of alcohol. Production of blue-brown dyes for coloring fabrics. Dwarf lichen Letharia vulpina. Litmus used as pH indicator made from Rosella species. Used in the production of perfumes and cosmetics. Avernia prunustri Lobaria pulmonaria, and used in the tanning. Lichens as pollution indicators Lichens are very sensitive to atmospheric pollutants such as sulfur dioxide. They are unable to grow in towns, cities, around industrial sites such as oil rainfairs and brickworks. So, the lichens can be used as a reliable biological indicator of pollution. By studying lichens on trees, a quantitative scale has been devised for the estimation mean of sulfur dioxide level in the given season. Thus, lichens are used as pollution monitors. Ecological roles. Lichens are significant in ecological succession. They are pioneer colonizer on bare rock surface. Crustose lichens are first organism to colonize bare rocks. Biological weathering of rocks by disintegrate rock into small particles by action of the acid. They secrete carbonic acid, oxalic acid. Soil formation favors the growth of the small rooted plants on rock surface. Thank you. Happy learning.